Why it is bad for the PCR? Someone tell me why it is not good for the PCR. It's just forming the primer dimer. Why it is not good? John, why primer dimer formation is not good for PCR? ক্লাসে তো অনেক কথা বললাম, এই হলো অনলাইনের যন্ত্র।
স্যার ডাইমার ফর্মুলা প্রাইমার তো কাজ করবে না, লাইক it will have secondary structures, so it will not work efficiently.
No, if I say like if it is dimer, when you increase the temperature for every first step of every cycle, 94 যখন denature করবা, it is then denature and become কি বলবো single stranded. Then why it is a problem?
Sir, I think there will be non-specific amplification.
Higher temperature requirement.
Yeah, Shah Faisal was saying something.
Sir, this is not good because there will be non-specific amplification.
Exactly. Because of the primer dimer formation, just see, this is your forward primer. Everybody can see this? দেখতেছো তো নাকি? Okay. Like this is red color is forward primer, black color is reverse primer. After primer dimer formation, the primer length will be something like from here. So this is the complete primer dimer length. So half of the primer is forward, half of the primer is reverse. So this primer, it is like a double length primer or double length DNA. It will not completely hybridize or it will not perfectly find their complementary sequence. It may found this sequence, but as it is having this sequence also, it will not go for the PCR amplification. The same way vice versa. এখানে এই সাইড আর এই সাইড, এই ফরওয়ার্ড প্রাইমার রিভার্স প্রাইমার মিলে you are going to have a long primer. মনে করো 30 base pair forward, 30 base pair reverse primer. Now after having the primer dimer formation done, then the primer that you are having is 60 base pair. Among these 60 base pair, 30 base pair may bind, but the other do not bind and PCR not happen. Vice versa. So বুঝতে পারছো বিষয়টা যে primer dimer formation always ends up with almost no PCR because those primers are elongated primer, and those elongated primer are not useful to having the PCR done successfully. Okay?
Sir. মানে স্যার ডাইমার থাকলে কমপ্লিটলি হাইব্রিডাইজ হবে না, এইজন্য পিসিআর
এক্স্যাক্টলি ডাইমার মনে করো 30 30 30 বেস পেয়ার ফরওয়ার্ড, 30 বেস পেয়ার ফরওয়ার্ড, 30 বেস পেয়ার রিভার্স, and you are having the dimer formation done, then what will be the product? 60 base pair, and that 60 base pair only it will find 30 base pair complementary sequence and bind there, but the other 30 base pair is not binding, so PCR will not happen. Okay? Okay, Gallo, then how to stop the primer dimer formation? The cheapest way is to use or do the PCR preparation on ice so that the polymerase enzyme will not function and it will not make any dimer formation. Gallo. But the other thing that you can afford is to use hot start PCR. Hot start PCR is a good thing where we are using specific DNA polymerase. Specific DNA polymerase. One of the polymerase that you can name is Amplitech Gold DNA polymerase. This TM is for trademark. So it's a company which producing this DNA polymerase, and they name this polymerase as Amplitech Gold. It is now available from many different companies with many different names. The main principle or the main function of this type of polymerase is same, though it's provided by many different companies. What is the function? This DNA polymerase is not active when you are getting it from the company. এটা যখন তুমি কোম্পানি থেকে আসতেছে, you are not finding or getting the DNA polymerase as an active form. এই পলিমারেজটা তখন কাজ করে না। You have to make something else to make this DNA polymerase active. The thing that you have to do is pre-incubation of 95 degree, usually for 10 to 11 minutes. So whatever the DNA polymerase you are having as this different type of polymerase, that is hot start polymerase, you have to heat the polymerase or the solution that contains this polymerase at 95. So it's a high heat for 10 to 11 minutes. Like more than 10 minutes is essential to make this polymerase active. What happened before, or if you do not make, or if you do not heat this DNA polymer, they will not do the polymerization. There is something changed in this type of DNA polymer so that they are not functional until or unless you are heating this polymerase. So that's why this type of polymerase is also named as hot start polymerase, and this type of PCR is named as hot start PCR. So when you do the PCR preparation at room temperature for long time, no problem if you use this type of polymerase, because the polymerization will not start. So it's a good thing, but these are expensive polymerase, about 5 to 6 times more expensive than the normal DNA polymerase that we are using. So it actually, if someone or any lab can afford, then it is good, no problem. But usually most of the lab do not use this. They are using the other thing that we discussed, doing this PCR preparation on ice. That's why they can avoid almost like successfully avoid the primer dimer formation. But if you have this, you don't have to worry about working on like cold temperature or cold room temperature is good enough, and you don't have to worry about the polymerization. Just do everything, take the tubes, put on the thermal cycler or on PCR machine, start the PCR machine. And this PCR machine, if you are using this type of DNA polymerase, you have to confirm that first step, prothom step should be 95 degree for 10 to 11 minutes. Then from the next step, it is just as usual. এখন বলো, at 95 degree, when you make this polymerase active, do you need this polymerase to become active for each and every cycle? কি মনে হয়? Have you got the point? একবার যদি, if you make this polymerase active by first step by 95 degree for 10 to 11 minutes, do you need this activation for each and every cycle? What do you think? Okay, nobody think nothing. That it is not required. One time incubation or one time activation is good enough. After you make this active, it will active or remain active until and unless you are using it. যতক্ষণ use করতেছো, it will remain active. So just the first step, you have to consider the pre-incubation at 95 degree for 10 to 11 minutes. And it has something, as we know that it is not active at room temperature, or it is not active when it is delivered to you. It has something, some modification. What is the modification? The chemical modification is there, and this modification is in the epsilon amino groups of lysine residues. So this is polymerase enzyme. It is a protein. It should have many different amino acids. One of the amino acids could be lysine. I should not say one. There should have many lysine residues. Many lysine residues in this protein or in this enzyme. And all these lysine residues, the amino group is added with something. Amino group of lysine is added with something that we don't know what is there. That addition of something, or the moiety or anything there, make this amino acid inactive. এই জিনিসটা যে epsilon amino গ্রুপের মধ্যে, if you put something there, then the enzyme is inactive. So this is how they make the enzyme inactive. Just put something to the epsilon amino group of lysine residues, and then the enzyme become inactive. Then when you hit this, that group or that moiety remove, or that moiety depart from that epsilon amino group, the lysine becomes same again, jeta lysine chilo, the lysine become normal again, no additional group added now, then it is become active. So this is the simple thing. They add something at the epsilon amino group or the amino group of the lysine, and the enzyme become inactive. And when you hit the enzyme, that group depart, that group remove from that amino acid, lysine, and the enzyme become active. That is the thing. So it's a very simple way of making this enzyme inactive, and you also have a very simple way to make this enzyme active by just hitting. Everybody get it? বোঝা গেল?
মানে স্যার হিট করলে যে রেসিডিউটা এক্সট্রা ছিল স্যার ওটা মানে চলে যায়, তখন হচ্ছে হলো আমরা
Exactly.
করতে পারি।
You put something যার কারণে the enzyme remain or enzyme become inactive, যতক্ষণ তুমি ওইটা থাকবে it remain inactive. However, যেভাবেই তুমি পারো, the way is people are using here is to hit. If you remove that group, the enzyme become active again. এইটুকুই, আর কিছু না।
জি স্যার, বুঝতে পেরেছি।
Okay, so this is very good thing about you can avoid primer timer by having this DNA polymerase. It's very simple, very simple. Now the next topic is multiplex PCR. In our DNA fingerprinting cases, we probably already remember or discussed that we have to do at least 13 to 15 locus PCR. We know about the number 13 at least. So to do or to get the 13 locus result, if you are doing the conventional PCR, you have to do 13 PCR separately, one by one, or 13 PCR at once in 13 different tubes. So it is a long, or it is a like tedious work. It will cost you more reagent, cost you more labor, cost you more expense, everything, and more time also. So to reduce everything of this, one of the protocol people are using is multiplex PCR. In multiplex PCR, you can use the same template, single template, and you get information. You can get information or amplification from different location of that template, like 3 to 4 PCR together. একসাথে 3টা থেকে 4টা PCR তুমি from the same template you can get. So that's why it is called multiplex. Multi means more than one. So you can do 2, 3, 4, 5, even 6 PCRs. It is not like only 2. It could be 2, 3, 4, 5, whatever. Now you decide how many you want to do. Like for example, you have to do 13. And if you do at once by multiplexing 4 PCR together, so you just have to do like 3 to 4 PCR reaction at once. একবারে প্রতিটাতে 4টা করে, if you do 3, you can get 12 PCR done. Or if you do 4, you can get 16 PCR done. So it will reduce your time, energy, and costing. So that's why multiplex PCR is used, and it is very popular, not only in our cases, means DNA fingerprinting, but also many different types of research work. If your lab are engaged in huge amount of PCR performation, or if your lab has to do many PCR together, একদিনে অনেক PCR they have to do, they are always preferring doing the multiplex PCR. And in case of multiplex PCR, this is just a pictorial or graphical presentation. Like this is A is the template from here to here, and you want to amplify these 3 locations: locus A, locus B, and locus C. And then you have to use this template, use the other things, for example, the buffer, enzyme, DNTP, nucleus-free water, and you have to use for these 3 different locations 3 pair of primers. For each location, you have to use 1 pair, 1 forward and 1 reverse. So here it is showing 3 locations together. You have to use 1, 2, 3, 4, 5, 6. 6 primers or 3 pair of primers. 3 pair use করে you can do in the same reaction with the same template. You can do 3 PCR together. But you have to consider, you have to keep in mind the following things when you're doing the multiplex PCR. কয়েকটা জিনিস মাথায় রাখতে হবে to design a multiplex PCR. First thing is the amplified product size. ধরে নাও you have to do this locus A, B, and C all together. You have to keep in mind that the product size should be different. Like if you have all the product size A, B, and C has the same length, for example 500 base pair, at the end when you gel run or when you run the product or the PCR product, you will see just a single band 500 base pair. You will not get sure that which one is amplified, A, B, or C. It could be all, it could be only B and C, it could be only A and C, whatever permutation and combination you can think of, if the length is same. So you have to consider or keep in mind that when you are doing all these 3 PCR together, you have to have the product size of different lengths. For example, here you can see locus A is short, locus B is medium, locus C is most largest. So here you can see this one, just for example, it has a product size what, like 200 base pair, it is like 400, it is like 600, whatever, just get the number. The product size should be of different lengths, so that when you run the gel, if you see 200 base pair length band, you can say that locus A is amplified. If you see, for example, 400 size length band, you can say that locus B is amplified. PCR of locus B positively happen. ঠিক একইভাবে if you see 600 base pair length band, you can say that locus C is also amplified. So 200, 400, 600, 200, 400, 600 base pair, or you can get 3 band 1 by 1 by 1, then you can say that all 3 PCR happen perfectly. And if you like you get 200 and 400, there is no 600. So you can say that locus A and locus B is amplified, locus C may be not present. Whatever, you have to think about the length of the product size to do the multiplex PCR. This is the first consideration. Any question here?
স্যার যদি আমি এখানে যদি তিনটাই সেম লেন্থের হয় তাহলে মানে পিসিআর রান করার পরে জেল রান দেওয়ার সময় তিনটাই হলো দেখা যাবে সেম ব্যান্ডে থাকবে, যে 40 40 40 যদি আমি সাপোজ ধরি, তাহলে তিনটা আমরা বলতে ডিফারেন্সিয়েট করতে পারবো না কোনটা কোন লোকাস। স্যার এইটাই তো মেইন সমস্যা।
এক্স্যাক্টলি, এখন এটা যদি আমরা এইভাবে করি, উই ক্যানট ডিফারেন্সিয়েট, লাইক অল আর 500 বেস পেয়ার, 500 বেস পেয়ার বরাবর ইউ গেট আ ব্যান্ড, ওকে, you cannot say that it is from A or B and C or it is from all. এটা বলা তখন কঠিন হবে, okay? That's why you have to have length variation. এটা একটা গেল। আরেকটা consideration, the other consideration that you must keep in mind is the primer compatibility. You are using like here 6 primers together. 6টা primer একসাথে ব্যবহার করতে, 6 primers together. So primer compatibility is very important. Like we discussed last class, what should be the primer. We just discussed about 1 pair, অথবা forward and reverse primer should be something like this. Now you have to have all those considerations here also. Like any of these primers should not be complementary to each other. এটা হলো first thing. একটু যেন complementary না হয়. Now you have to consider 6 primers together. There, for conventional PC, you just have to consider 2 primers. Now you have to consider 6, or if you do 4 PCR together, then you have to consider 8 primers. They should not be complementary to each other at all. That is the first thing about primer compatibility. Next thing is the melting temperature. As we know that for a PCR, difference between the primers melting temperature should be less than 5. Here also same. ওইখানে ছিল just between 2 primers difference should be less than 5. Now you have between 6 primers, the condition is same. The melting temperature difference should not be more than 5. It should be less than 5. These two considerations about primer must be keep in your mind when you're doing the multiplexes. First one is primer. All the primers that are using here, in this case 6. If it is 4 PCR, then 8. If it is 2 PCR, then 4. So you have to consider all the primers should not be complementary to each other. No complementation. First thing. Second thing, among these 4, 6 or 8, whatever number of primers you are using, the difference of melting temperature should be less than 5. A duita consideration about the primer you have to keep in mind. First one is the length of the product. Second one is primer compatibility. বোঝা গেল?
স্যার একটা কোশ্চেন।
Any question?
হ্যালো স্যার, কথা শোনাচ্ছে?
হ্যাঁ
স্যার একটা কোশ্চেন ছিল স্যার, এখানে প্রাইমার কম্প্যাটিবিলিটির কথা স্যার যেটা বললেন, স্যার আপনি যে বলতেছেন যে মেল্টিং টেম্পারেচার ডিফারেন্স হলে সমস্যা, স্যার এটা কি স্যার অ্যানিলিং টেম্পারেচারের জন্য আমরা এটা বলতেছি যে তিনটার অ্যানিলিং টেম্পারেচার কোয়াইট সেম হতে হবে এরকম কিছু কি?
Exactly. If just like you have 6 or 8 primers together, the lowest melting temperature, just get it as like 45, and the highest melting temperature is 65. It's a huge difference. And we know that when you design the primer, you have to keep in mind that difference between the primers should be less than 5. So এখানেও সেই একই condition. You have 6 primers, lowest primer অথবা lowest melting temperature could be like 50, highest melting temperature among these 6 should be like 54, no more than 5. বোঝা গেল?
জি স্যার, বুঝতে পারছি।
মানে এখানে ওই একই শর্ত এখানে থাকবে। The primer should be the melting temperature between the primers. Now how many primers you are using is irrelevant. যত primary থাকুক, between them lowest and highest difference should be no more than 5.
জি স্যার।
Okay, this is multiplex. So multiplex is very useful, very popular. In a single reaction, you just have to add one template, same amount of buffer, same amount of DNTP, same amount of magnesium, same amount of enzyme, just desired amount of primer. If you're doing 3 PCR together, 6 primer. If you're doing 4 PCR together, 8 primers. If you're doing 5 PCR together, 10 primers. Whatever, just forward and reverse for each and every primer. So that is the difference, but all other things same. In the same tube, you can get 3, 4, 2 to 5 or 6, whatever number you can get. The PCR is done in a single reaction. So for like 15 PCR, you just have to do like 4 tube reaction. So it will reduce your time, reduce your costing, reduce your labor, everything. And at the end, you are getting all the results together. So this is called multiplex PCR, which is very useful in our DNA fingerprinting cases. এরপর আসো, the last topic about PCR is PCR inhibition. There are many different types of inhibitors present, and this type of inhibitors interferes with the cell lysis necessary for DNA extraction. For example, you have a sample and you want to go to the, first you have to do the DNA extraction, as we see first, that when this type of inhibitor is present, it will inhibit the lysis of the cell. So if you are not going to have the lysis of the cell, cellta jodi lysis na hoye thake, you're not going to get the DNA. So it is the first type of inhibitor which interfere with your PCR. You are not having any DNA, you are not having any PCR. That is one type of inhibitor or one type of inhibition. Next is inhibitors that interfere by nucleic acid degradation or capture. This type of inhibitor interfere by nucleic acid degradation, like the DNase. DNase, if it is present in your extraction solution, it will do the degradation of your DNA. So you are not going to have your DNA intact. It will be like fragmented, and at the end you are not going to have any PCR happen. The third type of inhibitor which inhibits the polymerase activity. It will inhibit the polymerase or DNA polymerase that you are using for the PCR. Its activity will be inhibited by this type of inhibitor, and this usually preventing the enzymatic activity by binding with the active site of the enzyme. Enzymes has active site. Where something bind, it will inhibit the activity of that enzyme. Any enzyme usually have active site. If you design something that can bind to that active site, you can do the inhibition of that enzyme very successfully. So there are many different types of inhibitors which inhibit your polymerase by binding to the enzymatic or active site of that enzyme. In the next table, you can see there are many different types of inhibitors listed. For example, heme, then melanin, polysaccharide, then bile salts, humic products, urea, textile dyes. These are the denim. These all are inhibitors for your PCR reaction. These are the source of this heme. Heme, the source is blood. Melanin, the source is usually the hair and also the tissue. Polysaccharide could be fishes, bile salts. Then humic compounds could be the soil. Urea, the source is urine, and the textile dye, usually those clothing like the jeans, blue jeans, or those clothing usually has this type of denim dye or textile dye. All these are inhibiting your PCR. So when you are having those different types of samples, you also have to consider this type of inhibitors which can inhibit your PCR. And when the PCR is going to inhibit your samples, the thing you have to consider how to protect your PCR reaction from this type of inhibitors. The first thing is, we discussed probably, you just dilute the samples. Whatever samples you have, first do the PCR and you are getting no PCR. So it is a problem. There may have inhibitors there. So you have to dilute the samples, for example, 10 times dilution or 50 times dilution. When you dilute the samples, you also diluting the inhibitors. So then do the PCR. Most of the cases you can find the PCR is going on very nicely. So this is the first thing you can do. Or after even dilution, you are not getting the PCR done, then you can think of purify your extracted DNA by different protocols. If you purify the extracted DNA, you can confirm that, or you can assume that your inhibitors are also getting purified or removed, and you can get PCR done very easily. Another thing people are doing is to use bovine serum albumin, BSA. It's an interesting thing. This is common for many different types of enzymatic reactions. Bovine serum albumin. What is this? It's just a protein. Bovine serum albumin, BSA. What is the problem? Like, you have your DNA extraction done. And in our case, we are using raw preparation. For example, chillax extraction. It's just a raw preparation where you may have protease. Protease থাকতে পারে. And this protease will degrade your DNA polymerase. DNA polymerase is a protein. If your extracted solution contains protease, that protease can degrade DNA polymerase that you are using for PCR. So you are at the end degrading all the DNA polymerase, so no PCR reaction is happening. To prevent this DNA polymerase degradation, or to prevent this DNA polymerase cutting, you have to use bovine serum albumin. If you add bovine serum albumin in the PCR reaction, like you are adding everything, the template, primer, buffer, the enzyme, DNTPase, magnesium. Together with those, you add a bit of bovine serum albumin. You can avoid, or you can get rid of that problem, the problem of protease, which will degrade your DNA polymerase. Now, someone tell me, how adding of BSA can avoid this problem? একজন বলো যে এই যে তুমি BSA দিচ্ছ, just adding BSA and you can avoid the protease to degrade your
Sir.
DNA polymerase. হ্যাঁ
স্যার এখানে বোভিন সিরাম অ্যালবুমিন দেওয়ার ফলে যে প্রোটিয়েজ ছিল সেটা এইটাকে অ্যাটাক করতেছে, ওটাকে করতেছে না আর।
Very good. So you just adding good amount of BSA, that protease will degrade your bovine serum albumin more compared to the DNA polymerase. Just say that you have 10 polymerase and you are adding 100 BSA, and there are like 5 protease there. Those 5 protease will find this huge amount of BSA as their substrate, and they will be busy on cutting this BSA, and your DNA polymerase remains safe. Maybe one is getting cut, but most of them are remain safe, and that can do your PCR reaction. So just adding this BSA will avoid. Get the terminology or get the word very carefully. It is not inhibiting your protease. It is avoiding your DNA polymerase from cutting by the protease. আমি মাঝে মাঝে প্রশ্ন করি BSA will inhibit DNA polymerase. Now BSA will inhibit protease function. No, BSA do not inhibit protease function. BSA do nothing with the protease function. It is protease which most preferentially cuts the BSA, as BSA is present in huge amount compared to the DNA polymerase, and thus DNA polymerase avoid the degradation by the protease. So এগুলো একটু মাথায় রাখবেন. These are tricky things. The BSA do not inhibit the function of the protease, but just avoid the DNA polymerase from cutting by the protease. So this is one thing people can think of to inhibit the PCR inhibition. First thing was like diluting the samples. If dilution is not successful, then purification of the extracted DNA. And also in case of PCR reaction preparation, you can add BSA. That can avoid the protease contamination.
STR typing kits কি স্যার?
জোরে বলো বাবা।
স্যার STR typing kits যেটা ছিল লেখা লাস্ট লাইনে।
কোথায়?
Sir, last line.
Oh, these are nothing. Common ingredient in most STR typing. STR is the short tandem repeat. The kit that you are getting from the company are usually called STR typing kit. STR means short tandem repeat that you are looking for DNA fingerprinting. এটা তো মনে আছে না? STR করার যে kit গুলা, the kit contain all the things that is necessary for the PCR. That's
Okay, sir.
Okay. And those are the inhibitors, and you can avoid those inhibitions by doing those things. Hello. এরপর আসো precautions. You have to have some precautions against contamination. First thing is you have to separate the pre and post PCR sample processing areas physically. Like if you have a pre-PCR room or pre-PCR place and post-PCR place separated, you can avoid contamination very successfully. Like one room, small room could be for only pre-PCR. Pre-PCR means where you are doing the PCR preparation. Then you have done with the PCR preparation. Now you are doing 10 PCR reaction. All those PCR preparation will be done in pre-PCR room. Having done with the pre-PCR, all the preparations are done, and you take out those 10 samples to the post-PCR room where you are putting your samples on the PCR machine and start the PCR. Then after having PCR done, you run the gel in that room and getting the result. That is called post-PCR room. So pre-PCR and post-PCR, if these are separated, you can avoid cross-contamination or this type of contamination, many different types of contamination very successful. That is one precautions you can think of. If you have space, if you have enough funding or everything, then you can think of this very successfully. Another one is equipment. For PCR, you should have all the equipment separated. For example, pipettes, micropipettes, that will only be used for PCR, not for other things. For example, you have many different types of work where you need micropipette. For example, DNA extraction, you need micropipette. For DNA quantification, you need micropipette. For many different types of work, you need micropipette. So if you are using the same pipette for all these different types of work, you are increasing the chance of contamination during the PCR. So what you can do, you can separate, at least for the PCR, a set of pipette only for the PCR. So that is a good thing or good practice. If you can afford, separate all the pipettes or all the pipettes that are necessary for PCR in separate dryer or in separate place, where only when you are doing PCR you can use those pipettes. You should not use the same pipette for many different types of work. PCR purpose, only separate type of pipettes. If you can use, you can avoid the PCR contamination or contamination. And also disposable gloves should be worn and changed frequently. That is a very good practice. If you are doing the PCR preparation, you should have gloves wear, and it should be like changed frequently. Means like you are doing first PCR, like one set of PCR preparation with one gloves, then the other set of PCR preparation with other gloves. These are just frequent changing of the gloves can reduce the chance of contamination. Another thing, like you can do the PCR preparation in a laminar flow hood if available. So these are usually called the PCR cabinet. If you have, you can do all the PCR preparation inside the laminar flow hood or in PCR cabinet, or you can use the aerosol resistant pipette tips. The pipette tips that you are using is usually the normal pipette tips or the yellow color or green color pipette tips. These aerosol pipette tips are having some like this is a kind of like cotton plug inside the tips. Just get a picture information about this. Aerosol tips to the day আমরা
স্যার ভিতরে একটা ফিল্ট্রেশন টাইপ স্যার, একটা ভিতরে কটনের মতো
তুমি হয়তো দেখেছো। হ্যাঁ হ্যাঁ, এই যে ইমেজে যদি আমি দেখাই তোমাদেরকে, এই যেমন এটা বেশ ক্লিয়ার। This one is a like a filter inside the tips. Each and every tips contain this filter, so it will prevent the aerosol contamination. Aerosol contamination যদি তুমি prevent করতে চাও, you can use this type of aerosol pipettes, and it is much or very expensive. So only for PCR purpose or this type of purpose, you can use this type of aerosol resistant pipette if available or if you can afford. Then the reagent should be carefully prepared to avoid all these things are very common practice and very common good practices. And ultraviolet or UV irradiation of that laminar flow hood or PCR cabinet or the bench you are working with is kind of a mandatory things. If you have this type of cleaning process for your workspaces, is very good. Or you can use isopropanol or 10% bleach solution to ensure the extra DNA molecules that could be present there before you start working. You can avoid these things by using this laminar flow with ultraviolet irradiation, or at least by this ethanol washing, isopropanol ethanol or 10% bleach solution washing can prevent the extra DNA or the other DNA that is present in that workspace. You can remove those by doing these type of practices. Okay, so these are the precautions. One is the laminar flow hood or PCR cabinet should be used. Then aerosol resistant pipette can be used. All the reagents should be carefully prepared and carefully separated for only PCR. And then you can irradiate or you can clean your workspace. That could be the laminar flow, that could be your working space, anything, the bench that you are working with can be cleaned. If it is laminar flow, you can think of cleaning it by ultraviolet radiation or UV radiation, or if it is just a working bench, you can clean it with isopropanol or 70% ethanol or 10% bleach solution, whatever you have. Before you start working there, you just clean it with this type of solution, wipe that working space. You can think of your area very clean and can start the PCR preparation. So these are the precautions during the PCR reaction or during the PCR processing. এটা গেল একটা, আর হলো এটা যেন কোনটা, এটা PCR inhibition সম্পর্কে বললাম, then precaution গেল। তারপরে কি, these are just nothing. Advantages of PCR with forensic. All the things that you have are advantage. Compared to the RFLP, PCR is always advantage. The only thing, the contamination of DNA or like the contamination from other human DNA. If it is there, even PCR has nothing to do. Like you have a contaminated DNA of two different humans together. It is not the error of the PCR. PCR will give you the STR profiling or the result for each and every locus. A weird result. For example, two samples or two humans DNA together. For a single locus, you may have like 4 reports. 4 report means one is like 411, another one is 1012. So 4, 10, 11, 12. It is not disadvantage of PCR. I think it is the your or what I should not say drawback, or I should not say like which is your discompetency. It is whenever you are collecting the sample, if it is contaminated, you have nothing to do. It is from the source, it could be contaminated. So, but other things all are advantage. Just read it through. It's very simple. So these are the part of the PCR that we have just finished today. Good enough for today. And what we are left for the baki. Stop the recording.